Well, thank you so much, Abby, for the nice welcome. It's great to be here with everybody tonight. I'm really excited to talk to you guys about this project that we've been working on in Northern Lynn Canal for the last 6 years. So to introduce myself, I'm Anthony Crupi. I'm a father, I'm a sailor, and I love it when I'm sitting along the edge of a salmon stream in the autumn, in the rain, listening to the sound of gulls and eagles, smelling the decay of fish, listening to the sound of moms jumping into the water, teaching their cubs how to catch salmon. It's really been one of the more fascinating things I've seen in Southeast Alaska, and it's one of the reasons I've kind of dedicated my life to it. I currently work for the Department of Fish and Game as the research coordinator in Southeast Alaska, and welcome to so many people online tonight. From what I can see, you're watching from around the globe. Reading through the registration list, I wish I could give a shout out to all of you that I haven't seen in for so long. And to those who I haven't even met yet, thanks for your support and for your passion for bear conservation. Yesterday briefly, and we'll get to the next slide and then it should work just fine. So for a general overview of tonight's presentation, I just want to give you a little bit of a historical perspective on counting bears in Southeast, some of the research objectives that we have for the current project, display some of the bear movements that we've seen over the last 6 years, and then really dive into how we estimate the population and briefly touch on denning ecology. So I'd like to begin by acknowledging and giving respect to the Tlingit people for the land on which we gather and have had the privilege to work on and study. As a quick orientation, this is a map of Alaska, which is a state that covers a vast and varied landscape. It's about 663 million square miles. It's so vast, for instance, today it was 39 and raining here in Juneau, and 2 degrees with a blizzard warning up in Utqiagvik. 30 years ago, I graduated from Duke University and came to Alaska seeking wilderness and opportunity. For the past 25 years, I've been studying brown bears in Southeast. And this is where we're going to focus the discussion tonight. Relative to bears, grizzly bear density in Alaska varies regionally, with higher densities found near the coast, where in those places bears have greater access to salmon, and in these places we'll refer to them as brown bears. So Fish and Game's mission is to conserve and enhance Alaska's wildlife and habitats while providing for a wide range of public uses and benefits. Consistent with that mission, the department has been conducting research in Southeast for over 50 years. I need to acknowledge those before me who pioneered this research in Southeast, and this map provides a great overview of kind of the glacially influenced terrain and marine barriers that affect bear populations in Southeast. Outlined are the 9 study areas and the time periods where we've conducted research, and we gained a tremendous amount of information about brown bear ecology, habitat selection, denning requirements, as well as the population parameters that we use for management. Tonight, we're going to focus on the area here in red, Game Management Unit 1D. So why do we study bears? We're required by the state constitution to sustainably manage wildlife populations. But the reason why we focused on this area in Northern Lynn Canal is that in 2016, the Upper Lynn Canal Advisory Council sent a letter to the Board of Game requesting that the department study the brown bear population in 1D to determine the abundance, composition, and sustainable harvest rates. In 2018, we began that study, brown bear population density and habitat selection on the northern mainland coast of Southeast Alaska. This is a view from the upper Chilkat River looking down towards the ocean. So the purpose of this brown bear project is to collect the biological data necessary to manage for sustainability. Using GPS collars, we're investigating spatial relationships like home range size and habitat selection. We're collecting population demographic data such as survival, reproduction, and identifying sources of mortality, which are kind of critical to understanding how the population grows or declines. We're examining denning ecology as winter recreation activities continue to expand during this critical time of the year. And we continue to study population genetics in Southeast as the unique geologic and glacial history impact gene flow, and result in distinct population structuring. Ultimately, to manage the population, we need robust estimates of density and abundance, and how we do this is going to be the primary focus of my talk. So to give you a general timeline of where we've been for the last 6 years, we started this project in 2018. That first year, Stephanie Sell and I were able to capture and radio color 21 bears. In 2019, we colored an equal size sample, 22 brown bears. You can see along the bottom of this timeline that from the get-go, we started with a stratified camera grid array. And with that array, we've been able to really look at different things in the population where we can monitor the females with cubs that have collars or ear tags, and we're able to track a lot of this type of information because of this camera data that supplements the main course of our work. So 2020, everyone remembers, was a dramatic year, and the pandemic led to increased bear sightings, which starting in April and May and June, we really were getting a lot more people that were noticing bears around, and bears were right at the beginning starting to kind of get into some things. I've got this note there about the Y2K food. So there was this one place that they had been storing 55-gallon drums of rice and other food for 20 years, and bears ultimately found that, and when they have access to easy resources like that, it starts to lead to a problem. During the course of that year, we were able to GPS radio color 29 additional bears. Well, as summer progressed, typically what bears will, come July, start to feed on salmon. Well, that summer, the salmon were virtually non-existent. Bears are very adaptive, and they really can focus on a lot of different types of foods. And so, if there are decline in one type of resource, there's often things like berries that can sustain them through the year. Well, that year aligned also with a decline in the berry production. So ultimately, what that ended up with was about 50 brown bears that were killed that year through Defense of Life and Property killings, as well as typical harvest. So we knew right then that things were pretty serious. It was a 15 to 20% decline. We didn't know what the population estimate was at the time. We were fortunate that we had begun this study already, and our plan was to estimate the population in 2021, and so that's ultimately what we started doing. So that spring, we came up with a management plan. How do we recover this population and continue to study it as we go? So in that summer of 2021, we started catching hare for the population estimate, moved into August, continued catching hare. Camera grid array is still out. Fall of 2021, by the end of the survey, we had captured about 5 pounds of bear hair, and then it was a matter of getting all the hair ready and sending it off for kind of the DNA results that we need to make these estimates. We were able to collar another 8 bears that fall. 2022, we were able to continue this sampling of bears with another 19 collars went out. 23 was the last year of collaring, and we captured and collared 4 different bears. We're going to get into some of the details on what some of these collars do for us, but over the last year and a half, we've been working towards this population estimate, doing all the biometrics. We've recovered all the collars. There's still about 22 bears in the study area, 62 bears in the study area that still have some sort of marking with ear tags. And over the next year, we'll be working on all the publications and reports that have been generated from this project. So where did we start? Well, one of the key pieces of information that we needed was the GPS radio collars. And from those collars, we get a wealth of information. That totals up over the course of the study that we captured 91 different bears, 57 females, 34 males. We were able to distribute our capture efforts to various age and sex classes with 46 adult females and 11 subadult females. We collared 20 adult males and 12 of the 14 captured juvenile males. So the results of that collar data are illustrated in this map here. We have to recover each collar to download the data that's stored in the internal memory of that collar. Each collar collects a location at 20-minute intervals. It also records a temperature and an activity. You can see some of the wide-ranging trips that bears made over the course of this study. You can see a couple of wide-ranging movements here where 7 bears made transboundary movements into Canada for berries during early summer and always returned back to the primary study area during salmon spawning season. So you can see total collared animals, 87 bears, 32 were males, 55 were females, and almost 1.2 million locations. That here we break it down into males and females, where males are in this green color and females are in the magenta color. And you can see in some places where there's the green, the males are on top, which obscures some of the female locations in some of these high density areas. But essentially, we use these data to estimate home range sizes, where male home range sizes are more than 2 times that of females, about 128 square miles for males, 45 square miles for females. So as we look through the mortality of bears in this population over the last 10 years, this is just kind of a little illustrative graphic to show you how significant that event was in 2020. In 2021, the management plan went into place. You can see that we've been able to control the parts of mortality that we can for the last number of years. In between 2010 and 2018, there was a really high female proportion in the harvest. One of the reasons why we were asked to begin this study. 2020 was the highest mortality on record, and in 2021, reproduction was quite low. When we look at some of the sources of mortality in GMU1D, our research bears have accounted for about 32% of the total bear mortality. More than half, about 53%, has been from human harvest, and 46% of the mortality has been of females. Understanding these causes of mortality helps us understand more about the population and identify the sources of mortality that were previously unknown, such as bears that have killed other bears, as well as illegal take. In any given year, about 20 to 40% of the mortality has been of study animals, whether marked or radio collared. So how are we going to count bears in this dense rainforest? You can see these photos illustrate what the canopy cover can be like from above, and there's great challenges to trying to count elusive and cryptic animals that work under the cloak of darkness and live beneath this dense forest canopy in the coastal temperate rainforest of Southeast Alaska. So this isn't a new problem. Scientists have been working on this for nearly a century. In 1932, Frank Dufresne and Jay Williams began trying to count bears on the Admiralty, Baranof, and Chichagof Islands, where they would count the tracks along salmon streams. They started on Admiralty, and they'd measured the width of the bear track in the mud across the toes to try to distinguish between different bears. This ranged from 3¼ inches for cubs to 8½ inch for old bears, and they believed that they could recognize the same track if they encountered that track in other places. In 1938 and 39, they repeated this survey on Baranof and Chichagof Islands. You can see a map here of Baranof Island, and if you zoom in, you can see, okay, there were 3 bears in this watershed, 21 in another one. Again, this was repeated as we approached statehood, so in the late 50s on the ABC Islands. Well, as you're all probably scratching your heads like, yeah, that might be really pretty hard to do. And there could be a lot of biases, and depending on the substrate and how far you can actually get up into the salmon streams and whatnot. And so in 1981, Laverne Beyer and John Shane started on a new approach to try to estimate the population sizes for Admiralty and Chichagof Islands. And we called it a replicated capture mark resite, where we would capture a bear, make it really visible with a collar and these kind of fluorescent ear tags, and then we would fly surveys, multiple surveys between June and July to kind of repeat the number of animals that you see that are marked versus the ones that aren't marked. And using some fancy math, we were able to produce a population estimate and a density estimate for these islands. Essentially, that was the groundwork that laid our estimation techniques for across Southeast. Well, if it's not Admiralty, say Northern Lynn Canal, well, we know it's not as good of habitat as that. So what about a half a bear male isle? And so that's about the resolution of which we've been able to manage populations since that time until this current day. So to bring you my personal experience of where I started counting bears, was for my master's thesis, where I spent thousands of hours studying bear behavior. For 5 summers with a team of observers, including my wife, Lori, we identified individuals by their unique coat color patterns and the distinguishing marks on their hides. And we counted roughly 20 different bears each summer from this elevated tree stand along this one salmon stream in Southeast. As you can tell, that's a really small sample size when you look at the size of the state and Southeast in particular. So I'm going to introduce you to a bear here that I'm going to call right now Bear 27. For a number of years, we've watched this bear raise her young along the river. You can see that she's not collared, but she is distinguishable, and we'll return to her in a little bit. This was a photo of her from 2021, where she had 2 cubs of the year. So the grand question, how do we estimate bear populations? How do you count brown bears? How many bears are there in Northern Lynn Canal? So we decided to use a common technique that has been used over the past 25 years, where we estimate bear populations from catching hair and identifying the individuals. Once we collect that bear hair, we can then extract the DNA, and using microsatellites, which are sort of short repetitive sequences of DNA, we can identify each sample to the species. Is it a brown bear or a black bear or a non-bear species? Is it a male or a female? And all the way down to the individual bear that it is. When I first thought about naming this talk, I couldn't help but thinking of the Counting Crows album, Recovering the Satellites. And I thought to myself and kind of giggled, well, what if I call it Counting Bears, Recovering the Microsatellites? But really, Lori was the only one that was laughing at my stupid puns. So how are we going to catch this here? It's over 1,000 square kilometer mile study area. And fortunately, we had started 3 years before, and we had collared about 40 animals that we had received the collars back from. And so we had a little bit of information going into this. These are locations, again, where the males are green and the females are magenta. This was the information that we had leading into that summer of the population estimate. So created a kernel density estimate, which you can see here for just the months of July and August outlined in yellow. And we like to have these sort of stratified, kind of random ways to sample the landscape. So you can see this grid that's overlaid over the study area. But there's some places where we really are going to want to focus our effort, and that's what these location data show. So how are we going to catch this hair? Well, we've deployed 3 different types of detectors to maximize kind of our bear hair collection. Much like painting a bridge, as soon as we get through checking this trap array, we have to go back to the beginning and start it all over again. These continual checks are important to minimize the degradation of DNA in the sample and to maximize the number of animals that recapture, which are critical to the estimate. Here you can see that I'm going to describe in more detail the corrals, the rub trees, and the snares, which are the 3 techniques we use to catch the hair. So this is what most studies use, a scent-baited corral. And so we use 2 strands of barbed wire. You can see there's a high wire and a low wire. Well, we do that a little bit differently than other studies. We wanted to be able to make sure that that our estimate includes all age classes. So the lower wire is really there to sample cubs. You can also see on a big female like this that it also is able to kind of snag some of their lower hairs. And so the bear goes in to investigate this scent lure. It's basically a crab bait container with a really stinky lure. We would change the lure each time that we would go back and visit and collect the hair along the corral, and so that it was sort of novel and they would want to come and investigate it. They could get used to the scent and be like, oh, well, there was no reward out of that last time. I'm not going to bother getting through this barbed wire this time, but it's a very useful and productive way to catch hair. One of the downsides is that you end up with a bear comes in one side and goes out another side. Well, now I've got 2 samples at least from that same individual, which can be costly. Rub trees are another great way. Both brown and black bears rub on trees as a potential form of communication or an additional way to help shed hair. Sometimes maybe it just feels good. We located 20 of these marking trees while we were looking for our corral sites, and we were able to wrap a short piece of barbed wire around it so that we would catch hair on those. And with corrals and rub trees, there's this potential for a sex bias where, maybe males seasonally use rub trees more often. In some places, females with cubs may be too wary to go into a corral. And so the bear has to decide whether or not it wants to investigate or rub on these objects. And there's that cost of having multiple samples from both of those type of detectors. So fortunately, we had the great fortune of working with Laverne, who is an inventor. And he invented this single catch breakaway hair snare. That's essentially a wolf neck snare that closes to a piece of rubber. And as it cinches down onto the bear, it puts a little bit of pressure on, and that snags the hair sample. And then the piece of rubber breaks as it puts enough tension on it. We place them along bear trails, and they're kind of a more passive and less biased detector. Here's a photo of a bear encountering a single catch hair snare. It's then passed through and left a quality sample. This bear was determined to be a male, and he was recaptured 3 different times, as many as 14 miles away from where this hair snare was deployed. Here's a little video of what it looks like, bear going through it. So there it goes. The hair gets hung up. It's kind of nice because sometimes it'll get hung up on brush and it'll dry. And the weather and the rain really can degrade the DNA. So it's great and important that we go back and check these very often. So ultimately, we overlaid this. It's an 8 square kilometer, roughly 5 square kilometer grid, 5 square mile grid that was systematically placed. And there's one of those corrals in each one of these within the focused area, but we needed more hair. So we wrapped this hair around the rub trees, and then using this kind of prioritized area, we were focused on where we wanted to put the hair snares. We didn't want to put them in places that were not going to be worth our time. So it was really useful to use this collar data to guide where we should spend our time looking for trails. So ultimately, the grand day came. It was July 1st. We started setting out all of these hair traps. We ran the study through August 30th. During that time, we conducted 10 5-day sessions. We maintained 366 hair traps. 51 of those were the scent-baited corrals, 20 were rub trees, and we deployed 295 single catch hair snares. Here you can see that the corrals are outlined in that green color. The black triangles are the rub trees, and the white circles are where we deployed the snares. This was a huge effort. We couldn't have done this alone. It took two-person crews, so 4 people were on the project at all times. Over the course of the study, we had 8 different people help on the effort. We worked 75 days straight, setting out the traps, collecting the hair, processing the data. We had 53 cameras on these corrals and a couple on the hair snares. We used one boat, one truck, an R-66 helicopter, and by the end of those 75 days, my Fitbit logged to 450 miles. In the end, the session we successfully collected 1,267 hair samples from both black and brown bears. 790 of those samples were snagged at the corrals, many of which were duplicates and some were black bears. 73 samples were collected from rub trees. The hair snares accounted for 404 samples, and all but one of those in the hair snares was a brown bear. Just one was a black bear. This is a great, you can see in some of these corrals that the bears not only wanted to investigate what was hanging from the air, but as that lure would drip onto the ground, they would really come into the site and start to roll in it. And I perceive that they're doing this as a way to sort of mask their scent, much like some of your pet owners may have your dogs roll in stinky things certain times of the year. Well, bears were doing that a lot. We also had other species come to these corrals like moose. They would really be interested in the scent. So ultimately, we had all of these samples genetically analyzed. Many thanks to Wildlife Genetics International for providing the genotypes and handling thousands of samples over the years of this study and previously. The DNA results came back, and we had 434 detections that were successfully genotyped as brown bear, and we identified 148 unique individuals. 2/3 of those individuals were females, similar to the proportion of hair samples collected and the proportion of bears collared that were female. 72% of those bears captured were captured multiple times, between 2 and 27 repeated detections, which really helps refine that precision of the estimate. The hair samples at the corrals had a similar, at the corrals and the hair snares, they had kind of a similar genotype success rate, which is important. So when we look at the black bear samples that we collected, we were able to collect 128 samples that were identified as black bear. That broke down into 63 individuals and 77 unique detections. With that, a limited level of recapture, we're going to still work through the data to see if we have a way to produce a reliable estimate. Just over half of these black bears were females. And so here you can see if this is a higher elevation corral. This black bear is coming into the corral. You can see it's getting raked by this barbed wire as it goes through. And then as that goes in, you can focus in there and see on those middle two barbs that it's snagged a big sample of hair. And that's kind of how these things work in collecting the hair samples. So these are the bear movement paths that were collected during the same time period that we caught the hair. It represents 24 different individual bears and 82,000 locations. And you can see one that, boy, it really looks like the hair traps were put in good places. We really didn't miss many places where bears focused their activity. We had places that were kind of sampled appropriately, but it also shows kind of some of the wide-ranging movements that bears made. So some of these bears would encounter a detector at the southern end of the study area and then all the way up into the middle of the study area. And so this spatial component is really an important part of the model, as well as we're able to incorporate these telemetry data into those estimates. So I just wanted to look a little bit at, well, which corrals really did well. So this is just the corrals. And if you see a bear facing to the right and it's outlined in green, those are places where we successfully collected and genotyped brown bear hair. And if it's facing to the left, those are some of the corrals that did not, they maybe produced brown bear hair, black bear hair, but not black bear samples, not brown bear samples, sorry. Here's another figure that shows the number of unique individuals that each corral site. So as well as the rub trees and the single catch hair snares. So you can see in some places where density probably wasn't as high, we identified fewer individuals, and in other places there were 8 to 11 individual bears that were captured at that particular trap. You can see some of the bears tried to get away from us any way they could. This one was trying to do the limbo to get out from being snagged by the hair. Not really. She was really rolling around and using this wire to really dig her back even more into the masking her scent with this putrid lure that we use to attract them to the site. This is a graphic that shows all of these movements of the 24 different animals during the course of the study. You can see the time along the top and the wide-ranging movements of these bears throughout the study, just in July and August of 2021. So in the end, we estimated the population density of brown bears using genetic mark recapture with a maximum likelihood method implemented in the seeker package in R. That stands for spatially explicit capture recapture. So where the trap was mattered, where the bear was recaptured matters, and essentially we work on the precision of this estimate by the number of recaptures that you get. So if by the end of the study every bear was still a new bear, well then that estimate doesn't have a lot of reliability, and there's really would be a wide confidence interval around that estimate. But here where we've recaptured so many individuals multiple times and over wide-ranging movement patterns, we were able to really get a refined estimate. So the top model that was selected included the telemetry data, which really helped improve the range parameter, and sex was also included in this top model, as well as the baseline detection probability and the range parameter. So we included sex, trap elevation, and the type of the trap really were important to creating the best model. In the end, the density of bears in this study area was 108 bears per 1,000 square kilometers, plus or minus a standard error of 11, and a population size was 307 bears in Game Management Unit 1D, plus or minus 31, with a confidence interval that ranges between 258 and 365. So there are a lot of advantages to this system that we used of catching hair and using the telemetry data. The telemetry really improved the precision of the detection probability, and it substantially increased the female parameters. The hair snares were also a really great advantage that this study presents, and we suggest that other studies use it moving forward. One, there's no mixed samples. It catches one sample from an individual bear, and then it detected 2.5 times as many individuals as if we were to just use the corrals alone. It's a relatively unbiased detector, so females that may be less likely to use rub trees or the scent-baited corrals, they're just walking down a trail and come through this single catch hair snare. So no choice really has to be made for them to be sampled. And it greatly increased our estimates of abundance and as well as the precision. So back to bear 27. Why is it bear 27? Well, this is the bear that we recaptured 27 times. And you can see here, this is a spring photo before we started the estimate. And fortunately, this bear has been known and photographed. And so as we look at some of the data that this bear provided to the study, she left over 40 different hair samples. Outlined here in magenta are the traps within her home range that she encountered and left a sample where it was successfully genotyped. That sometimes it would be during the same occasion. We just used that one sample for each occasion at each trap. So this led to 27 different detections. She visited 23 different traps, which included 16 hair snares, 4 corrals, and 3 rub trees. From her DNA, we learned some other things. So here are a couple of the corrals that she visited. She visited this one in the upper valley, and you can see she's kind of stepping over this top wire. Well, it left a really good sample on that top wire. Her cubs went in, they got caught on the lower wire. So we now know from the genetics that this bear is the offspring of another bear that we had captured back in 2018. We were able to run her pedigree analysis, and that's a 15-year-old female along the Chilkat River. This bear is a direct offspring of. We were also able to identify from this genetics her cubs. We learned that one of these cubs is a male, one of these cubs is a female. One of them looked like it was often not really following mom's directions that well and would run off. You could see in the photos to where it was there 1 minute and gone the next. And sure enough, I was able to track it down to the lower wire on the far side during the 4th check. And that was the male sample. And he was quite precocious. We were also able to identify that this female was a half sibling of another bear that we've have marked and collared, bear 902. So it's really interesting what some of these genetic data can show us when in the absence of having our more traditional collar data. So an assessment of the long-term reproductive potential of brown bears is essential when we evaluate kind of some of the factors that are limiting population growth. And bears have a really low reproductive rate, and the recovery from population reductions is often slow. We can count the number of cubs present in each collared bear by seeing it from the air, seeing it on these cameras, as well as oftentimes we can identify the ear tag marked bears. We've tracked over 100 bear reproductive events over the 6-year period, and more than half of those times those adult bears were single without female, as you can see in the pie chart in blue here on the right. 2 cubs, when they did have cubs, was the most common, and triplets were found in about 8% of the females. In 2020, you can see in the figure on the left, the average litter size was down to just 1.5 cubs. And the following year, the percentage of females with cubs was really low. And we only, I think, had 3 bears that year that were marked or collared that we were identified that had cubs. So we knew that this population recovery and growth was going to be slow, and we estimated that it's probably going to take about 5 years to be able to recover the population to where we once were from after following the 2020 event. So to put all this demographic data together, our education staff and biologists combined reproductive data and the survival data to illustrate kind of the reproductive capability of, say, one female. Could be bear 927 or bear 27. At the first stage of reproduction, on the top line, you can see a 5-year-old female that has 2 cubs that stay with her for about 2.5 years. Half of those cubs survive to adulthood, continue this pattern throughout her life. She would, after about 20 years, she would produce 4 bears that survive to have offspring of their own. That would be the best case scenario, given that the interval between cubs is often greater than 3 years, and the average age of female mortality in 1D is about 10 years old. But it's really a nice way to be able to illustrate kind of the reproductive potential based on the survival and reproductive data that we've collected during this research. So why does it matter? Ultimately, we can pull all of this data together to track the population over time. And what you see here on the top in the blue line is the population size. And you can see the dramatic effect of the 2020 event in green, where we go from an average of 12 to 16 bears sealed each year to about 50 bears in year 2020. Following in 21, the management plan started where we're trying to keep it at a lower level. And you can see with that effort that the number of females in the population is predicted to, right now it's current data up till 2024. It's staying steady, slowly growing. And as we project out into the next couple of years, if we were able to maintain this level of mortality and reproduction and survival, a lot of unknowns and kind of question marks in there, but this is our best case scenario for how we can build the population back and have some idea of what these estimates do to make that happen. So I said I'd briefly speak about brown bear denning. Most often this time of year, I get the question, when are the bears going to finally go into the den? And so this is a figure here on the top left that shows the bears that we've had collared. We can tell when the bear goes underground, the temperature goes up on the collar, the activity really slows down to next to nothing, and oftentimes it'll lose connection with the satellite. So based on that, we're able to tell when the bear enters into the den, and likewise, we can say the same when the bear exits the den in the spring. And so I tend to think that it's usually around Thanksgiving is sort of the median date of den entry. But you can see here, as you get into the second week of December, there aren't too many more bears that are still left up. Males are going to be the latest ones to, on average, enter in the den, where it's later in November. Females with cubs would be the earlier cohort to go into the den. I did one of these virtual wildlife series 2 years ago. There's a QR code down on the bottom if you're more interested in denning. That'll show you the whole talk and really dig into the details of denning in Southeast, which I find really fascinating. And so what kinds of future directions can we use with this catching of hair and other types of DNA? Well, we know that it's useful for estimating populations. As we get into more genetic tagging, it's not just hair that we can use. We can also, there have been studies that have used saliva off of salmon that are swabbed, where we can get the saliva of the bears on the salmon that have been captured and partially consumed and get information that way. We can also find bear DNA in feces. And so this was just one way that we've estimated the population. There are a lot of different ways that other people have started to investigate population size and density. Another future prospect is using cameras, possibly in combination with this DNA, to where we can estimate population and even density using some of these camera tools. And ultimately, the question is, do we need to get into more genetic monitoring? Can we detect these changes in population size over time from genetic tagging? Can we determine some of the growth rates? There are a lot of neat developments in the wildlife ecology field where we are starting to find that we can learn some of these things. We can learn different demographic information about their survival and reproduction, and using new types of models like an open population spatial capture, recapture model. We can really tell a lot of different things, as well as connection to other populations and movements over time. It's really an exciting new field that we have ahead of us, as well as harvest management. Are we able to come up with harvest rates based on, we collect a tissue sample each time an animal is harvested, and so can we see where we've seen that animal over time and really use that in towards detecting these changes in population size and kind of the genetic diversity as that changes through time. So thank you so much for being here with me tonight. I have to reiterate and acknowledge that this is very much a collaborative project. It wouldn't have been possible to have done this alone. I'm indebted to a cast of biologists who came before me, and thank you, Stephanie, for all of your help and efforts. Congratulations to Stephanie, who will be filling my old role as bear research biologist in Southeast. Thanks to all the pilots and technicians and volunteers for all their time and effort and dedication to improving our knowledge of brown bears in Southeast Alaska. And thanks so much. Appreciate you all being here tonight.